Hello, and welcome to Review 1. And today's review paper we're going to be going over is named Prediction of Acquired Antimicrobial Resistance for Multiple Bacteria Species Using Neural Networks. And it is by Aytan-Aktug et al. Sorry if I mispronounced that name. I'm not good with names, I will admit that. As always with these types of videos, there will be multiple types of references for this paper down in the description if you want to go look this paper up more or reference it in your own work. To get started looking into the paper, we're going to start with the main points that are raised, so what questions this paper is trying to answer. Then we're going to go into the conclusion first, just to kind of bring the questions and answers together. And then after that, we'll go into the actual details of the paper, as well as what they did right, what they might have done wrong, questions that were raised, and as well as possible future work questions that can come out of this paper. So with that, let's get to the main point. So there are two main questions that are posed in this paper. The first one looks to ask, if you have an individual bacterial species training data set, and you have a separate multiple bacterial species training data set. So there are multiple bacterial species in one training data set for a machine learning algorithm. Which of those would be better at predicting antimicrobial resistance profiles or AMR profiles? So deciding whether that one bacterial species would be susceptible or resistant against any number of antibiotics, which one of those would be better performing at that task? Would the individual training data set be better at predicting AMR profiles, or would the multiple species training data set be better at predicting? So that is the first question. The second question looks to also use that multiple species in training input data set, and it asks then, okay, we have this data set. Can we then predict on novel bacterial species? Species that might have just been sequenced, they might have started going through the bioinformatics pipelines out there, and they might not have any AMR profiles yet, or very few. Could we predict on those to kind of get ahead of the curve, so to speak? And what would the performance on that be? So those are the two main questions posed in this paper. Now, the conclusions for these questions are, for the first question, it is that there was no real performance gain. However, there was no performance loss either. So adding the multiple species training data set and the individual species training data set performed similarly to each other. And that's not actually a bad thing. That means that it could be viable to actually have a larger training data set with multiple species, then be used to predict on those multiple species. So, for example, instead of having a single model per species, you could instead train one large machine learning algorithm model and then use that model for a multitude of species with no performance loss. So that is actually very good to have, because then that would save on training time, it would save on space to hold all the models, it would save on complexity. So that is a very good conclusion to actually have. It's not the best because it did not gain performance, but it's still good that it did not hinder performance as well. The second conclusion for this second question is that it could not actually reliably predict on novel species. So they found that there was very low prediction accuracy when giving it new species that were not in the training data set. So with that, it would be unlikely that you'd be able to predict on new species using the algorithms posed in, or the models at least posed in this paper. Now to start actually going through what happened with the paper, what steps they took to get to these conclusions. The first thing they did was they had to build the training data set. So they ran sequences through the two bioinformatics tools called ResFinder and PointFinder. PointFinder is a way to detect mutations in the DNA sequence, so SNP values, or single nucleotide polymorphisms. Then ResFinder was used to detect plasmid acquisition, or ways of bacterial species to transfer different possible resistance genes between each other. And then both these tools were actually combined in ResFinder version 3.0, so they used that then as a just combined tool. So the first thing they did was they went through and used PointFinder, ResFinder to then calculate a binary output, which is basically a way of saying, yes, the bacterial species has this resistance gene, or no, it does not have that. And then they also used then what's called a Blossom 63 matrix score. So a Blossom X matrix is a way of calculating a number for an identity, of saying it'll compare two sequences and it'll give an identity score to say how alike those two sequences are. So they use the Blossom 63 matrix score, and that's two different types of data input there, the binary and the score input data. And then they also used clustering techniques with assembly to collect amino acid information as well as nucleotide information. And then they lastly used a combination of binary and score. So you have these 5 different types of input. You have binary, you have score, you have amino acid, you have nucleotide, and you have the combination of binary and score. So you have these 5 types of training input. So the first thing they had to do was they needed to see, okay, which one of these is the best type of input for our algorithms. And so that was the question they posed. And so they looked at using a random forest machine learning algorithm with 5-fold cross-validation, and they used E. coli and M. tuberculosis for each input type to test which of those are the best. And they found out that both the combination of binary and score worked the best. So throughout the rest of the paper, they used that binary and score combination input data as the input for their machine learning algorithms. This next question that they asked was, could we train a neural network with this input feature set and then compare that with the random forest to see, are they similar in performance? Is one better than the other? And they used a paired t-test to look at the outputs to see, okay, how do they compare? And they found that they actually performed similarly. So the neural network did not perform any better or worse than the random forest. So with this, they looked at a problem that random forest had, and that is random forests are difficult on training. If you have input data, so if your classes or your output predictions that you want to predict on, if you have one that's not included in your input, then you will not be able to predict on it later on. And they were having trouble with this because there might be new profiles in the output that were not accepted in the input. And so with that, they then decided that the neural network would be better to use because then they would not have to worry about working with missing values for the output. So through the rest of the paper, they then decided to work on neural networks instead of random forests. And then they decided to say, okay, the next question is, they looked at a single species dataset versus a dataset containing all species. And they put those two different types into the machine learning algorithm, the neural network, using a paired t-test to compare them, and they found again that they were similar. The new multiple species dataset did not outperform and it did not underperform the single species dataset. So then their next question was, okay, they then have two ways to create this new multiple species dataset. The one method was aligning with a specific species, so each bacterial species that was in the dataset would be aligned to its own individual species. And then the second method would be to align each species with a concatenation of just general references for the different genes. And so they then tried creating the two different data sets using those two different methods. Again, they compared it using a pair t-test, and again, they found that there was no difference. So then they decided to use the concatenated, so method 2, for building their multiple species dataset. Then one last thing they posed was they looked at a one hidden layer neural network versus a multiple hidden layer neural network. And in their conclusion, they had that both seemed to have the same results. There was no difference between one hidden layer and multiple hidden layers. And then the last point for their paper is they wanted to see, okay, does PointFinder or ResFinder work better or worse than this neural network that they've come up with? And in that conclusion, with all that, they found that PointFinder, ResFinder did actually beat the neural network on 6 out of the 8 species that they tested on. And they came to that conclusion, and they made a note that is worth noting that yes, the PointFinder ResFinder is built on top of well-documented bacterial species and resistance profiles, so resistance genes, and so because of that, it will be hard to beat that. And then the two bacterial species that they did not beat were already hard to predict on, because it was multiple different types of mutations, and even just looking at the mutations themselves and genes does not always account for the resistance in those two species. So actually both the ResFinder tools and the neural network were not very good at predicting on those two species. However, the neural network just barely beat out ResFinder. So you would not be able to necessarily replace ResFinder with their neural network they created. That's the main conclusion that came out of that last step. So that was all that they did in this paper with looking, they went step by step in from what I just mentioned, and they looked at each different obstacle. They decided neural networks were best to use because of the less information, or being able to predict on missing classes. They did not actually mention which neural network model they used. They did not mention whether it was a one hidden layer or multiple hidden layer neural network model. They just mentioned that both had the same performance. And they also mentioned that they are using the multiple data set that is being created using binary and score combination data, and it is built off of concatenated reference databases for the different species. So with that is how they actually came from their question that was posed at the beginning to the conclusions that were posed at the end. So now that we've gone over all the steps, we've gone through the main points of the paper, the conclusions that were brought up, the steps to get from the beginning to the end, let's go over some things that the paper did right and some things that the paper did wrong. So one of the things that the paper did right is they used a paired t-test for every comparison that was made, and they had a p-value of 0.05. That's good because then you don't have the objectiveness of just posting the data, the information such as F1 accuracy, area under the curve, and just saying, hey, here's our results. We think they're the same, we think they're better because of blank. And so the paired t-test is just a way of saying this is the same or this is different because. So that's a good thing to have. The second thing is they did use area under the curve, which either area under the curve as a performance measure or F1 score as a performance measure is usually the best thing to do, because especially when you have imbalanced data sets, accuracy can be very skewed. And so you usually don't want to use accuracy as a performance measure. Either F1 or area under the curve are good performance measures for machine learning. And so, especially with bioinformatics and bacterial species looking at resistance with antibiotics, you will have a lot of data sets that are just naturally imbalanced. So that is good that they thought ahead and used area under the curve. And then once they moved to the last step and were comparing PointFinder and ResFinder, they did use a different measure, and they also included measures for F1, specificity and sensitivity, which specificity and sensitivity are general performance measures for bioinformatics. So that's good. And they also included an F1 score as kind of a backup for the different measure that they used, because in the last step, again, they were comparing PointFinder ResFinder against their neural network, which are two separate models. One is not machine learning, so it might not be possible to use area under the curve as much with that. So that's some of the things that the paper did right. Now, some things that the paper did wrong was that they used what's called an epidemiologic cutoff value, or ECV value, and they compared that against clinical resistance. And they did mention this in the paper, that the PointFinder ResFinder did use the ECV value when comparing resistance profiles, or giving resistance profiles, whereas the machine learning models use the CLSI clinical resistance, clinical breakpoint values, as well as some UCAST breakpoint values as well mixed in. And they mentioned that it'd be preferred to use MIC values to compare instead of these two different ECV and clinical breakpoint values, just because they are two different types of resistance providers. So that is something that they did wrong. It did not mention that they did switch over to MIC values. They did not mention that they had MIC values at all. In their paper, they mentioned that the data that they got all this from, the data did have MIC values, and then it also had the clinical breakpoint values, and that they also compared them. But it did not say that they only used the MIC values, or they only used the breakpoint values. So there's a little ambiguity there. And then another thing is that they mentioned that they used upsampling to combat the imbalance in the data sets. However, with this upsampling that they did it after they did their cross-validation partitions. And so that does not help the machine learning algorithm as much as if they did the upsampling and then did the cross-validation. Because if you upsample into the same partitions, then you have the same data being shown in the same partition, and it might not actually be spread out through the different partitions, which is what upsampling should be used for. And then on top of that, when they did their upsampling, they mentioned that they just upsampled and copied basically every element 5 times. And usually you don't just want to copy things, say X times. You instead want to do some adjustment to either the input or the output based off of the population of sample points that you have. And they never did actually mention what sampling technique or algorithm they used. And so that's also more ambiguity that's brought in with the paper. They should have said, this is the technique we used, and so then they would not have to necessarily say, this is how we upsampled. They could just say, we used this technique or this algorithm, and then had it copy 5 times, which would have been okay, but they never did mention that. And then that was this ambiguity problem was a general issue throughout the paper. They'd mention things and then never elaborate on them later on. So that was also just a general problem that was with the paper. So now onto the questions that came out of the paper. There's only really 3. One of them is, when they collected the data for the resistance profiles, were they able to obtain MIC values, or were they just able to obtain the resistance profiles, so the MIC values compared to the breakpoint values, and had a susceptible, resistant, or intermediate value calculated out of that? Were these values available or not? And then the second question is, what technique or algorithm did they use to upsample data? There are a few. I know I have personally used something in previous papers that I've worked on called Smotine. That is a Python library you can use. And so they did mention they used Python in here, so possibly they could have used this as well. They just did not mention it, but again, they did not mention it, so you cannot really assume anything of what they did and did not do. And then the third question is, how did they come up with these optimal parameters for the random forest? They did mention that they had the number of trees be 200 and they left everything else to be default, and that was turned out to be optimal. They did not go into why that was the optimal for them. They did not go into what was used to calculate the optimal. Same with the neural network. They said they used the rectified linear unit for the in between into the hidden layer, and then sigmoid activation function in the output. And they also talked about the learning rate, and they also talked about the multiple hidden layer option, but they didn't really go into why they chose the hidden layers, why they chose the neurons. They went over a little bit about the learning rate, but there was no real way of indicating which of those options were optimal, why, what tools did they use, if any, for coming up with those optimal values. And so those really are the 3 main questions that I have, at least, coming out of reading this paper. So now on to the final part, which is possible future areas of interest that can come out of this paper. So one of them is looking at MIC values and then trying to compare, using a machine learning algorithm that's been tested, compare the algorithm predicting MIC values for all those different bacterial species, and then have it also potentially either predict ECV values or compare different clinical breakpoint resistance profiles for the different breakpoint values. So maybe compare UCAST, CLSI, and those 2 breakpoint values and ECV using the MIC values that are generated from the machine learning algorithm, or have the machine learning algorithm predict MIC and those other values, and then compare all of them, which would be more viable to predict resistance, which could be a better means of accuracy. Would it be better to predict on one single type of resistance profile, or predict the MIC and then have it follow through with the clinical breakpoints? Which one of those would be viable? And that is actually something that I am currently working on with my colleagues. Another one would be looking at novel species in a little more detail. Now in the paper they did mention that they could not have decent accuracy, but they also only had 8 species, which is not that much of a variety. So potentially, if you could allow for a larger diversity of species and potentially different orders of species in the same data set, training data set, could that then predict on novel species, and how would that fare against PointFinder and ResFinder? So that would be something to look into. And then the third idea would be something that they alluded to at the end of the paper, which is that there's the potential for multitasking machine learning algorithms, or algorithms that can do multiple things. So for example, you could have an algorithm that does predict an MIC value as well as predict the resistance profile, or have it predict an MIC value and predict at the same time a possible resistance genes in a certain sequence. So there's the multitasking machine learning algorithm there. So potentially, could you create an algorithm that predicts on a MIC value, say a neural network, have it train on MIC value, and then keep the beginning part of that untrained, and then try and train a new output layer for, say, resistance genes? Could you then have machine learning algorithms that are multi-purpose? And so that would even break down the complexity even more, and possibly even provide different solutions. So instead of having resistance genes and just predicting on sequences, you could potentially predict on what could come out of the MIC value instead. So there's all those different things you could look into. And with all that said, that is the first review done, and the paper was very well written. It actually did have a lot of insight into a few different areas, such as using PointFinder and ResFinder as viable options for future research, and different types of input data, as well as different ways of setting up that input data. And it left the door open for future ideas and questions. And as I've mentioned before, the reference for this, as well as a link to a journal page that you can go and see the paper yourself, will be in the description. And if you have any suggestions for papers, preferably machine learning papers that you would like me to review, leave them in the comments, and maybe even discuss your own questions and comments about the paper if you were able to read it in the description as well. And with that, thank you for watching, and I will see you in the next review.